discs on them, and that antibiotic will diffuse into the agar with the bacteria, and then you'll see what happens. So that's basically what we're going to talk about today. I introduced some of those antibiotics and the whole idea of antimicrobial drugs. So here's our first kind of eye clicker question. So if you have your little eye clicker things, you can hold them up. This is just kind of testing your knowledge before we get into this. Okay, so everybody have, yep. Okay, so it looks like most people, so some do come from fungi, so you're right on there, but most of them come from bacteria. Okay? And so we're going to talk about why that is and what are they doing. So that's our main source. All right, so a little bit of history of chemotherapy, and chemotherapy is just kind of what the name sounds like. It's a chemical therapy to fight disease. And it began with Paul Ehrlich. I'm sure it probably began much before then, but it's just not recorded. During World War I, there was a very high need to have something to be able to protect soldiers when they came back. They had amputations, they had different things, and they would mainly die from infections of their wounds. So they were looking for something that would be able to inhibit the growth of the bacteria that was killing them. So they tried just about everything, and at this point in time, a lot of the individuals who were using dyes and stuff with materials from, let's say, cloth and things, they would turn these over to the German government, and then the German government would test all of these different reagents that they got. And they would find that some of them would actually be selectively staining organisms, and that's what gave them the idea. If we can selectively stain, we might be able to selectively kill. So this is really kind of this whole idea of being able to selective against different bacteria versus a human cell. And so this is Paul Ehrlich, and he's the one that had this magic bullet, and the magic bullet was that whatever substance he would be able to have a person take, it would harm the bacteria but would not harm the person. But we'll see in the next slide that that is not the case. This was what gave him hope. They had different bacteria, and you can see these little clearings around the bacteria. So this was showing that some bacteria were secreting something outside themselves that were killing other bacteria. So this made them feel if they could just isolate what these materials were, and then figure out what bacteria they were affecting, and then figure out what bacteria was infecting a person, they could give this person this kind of a chemotherapy drug. This is the selective toxicity idea, and this is his magic bullet. But of course, that's not exactly what happened. These are some of the things that he found, and you can see it's very hard to see, but this is arsenic, that AS up there. So that's arsenic. He had these rather large molecules that they synthesized, and these would be ointments and stuff that would be used to treat syphilis. And they were successful, but they were also really toxic to the individuals. So it did its job in one respect, but they realized that this was not the long term. And then they kept looking for different things, and this salvarsan was the first thing that they had that actually synthesized something that was antimicrobial, and this is the molecule that they ended up finally synthesizing. And then years went by, and they kept trying, and they looked at this red food dye that they had, or fabric dye, and they actually synthesized it, and this is kind of what it looked like. And so they were trying to build off that molecule and say, all right, let's try to design something of this nature that we could give to individuals. And it really wasn't very successful, but because of all this effort, they did find sulfur drugs. And this was found by Domock, and he got a Nobel Prize in 1939 for finding a substance that would help humans versus bacteria. And unfortunately, he could not collect his Nobel Prize because this was now World War I going on, or World War II going on, and he was not allowed to leave Germany. They thought if he ever left, he may not ever come back. So he was not allowed to go and collect his Nobel Prize. So they kept on with World War II. They kept on using these sulfa drugs, and they were successful. Didn't help as much as other drugs like penicillin. So we'll see that. So this is really not the sulfa drugs. These are not antibiotics because they're not made by one organism to kill another organism. That's what an antibiotic is. This is just a synthetic drug that is made to antimicrobial, we'll call it. And the first one that really was found that was extremely useful was by Fleming, and you probably all know the story about that. He left some plate out, and he found on that plate, when he got back from wherever he was, that there was a lot of growth that used to be here, but now it was gone. So they said this fuzzy thing right here was something that was secreting something outside itself that was killing all the bacteria. And this fuzzy thing happened to be penicillin, which is a fungus. And that was the first isolated organism that could produce a antibiotic. Okay, so this is one organism killing other organisms. Okay, and it's really interesting because Fleming was not a chemist, and he really didn't understand how to isolate penicillin. So it went for years and years, and from 1928, from the discovery till 1939, until we could actually purify it and use it. And what he did is he was gracious enough to contact other chemists. And he said, I have this, what I think is a wonderful discovery, and if you will help me with this discovery, at least be able to isolate it and synthesize it so that we could start using it. He'd already tested it on people, and it worked. But he just could not get enough of it. And he didn't know all the purification methods, so he got some organic chemists in here in England. And in 1939, right when we needed it with World War II, those chemists in England went to the United States because the United States at that time was not at war, and England was. So they gave the antibiotic to them, the penicillin that they had isolated, and of course Merck just ramped up all the ability to make a whole bunch of penicillin. And it was really a game changer. And there's a very interesting book in this. Talks about in England when they were being bombed all the time. These individuals that knew that penicillin was a game changer, they were afraid that the Germans, which were already in England, they were afraid that they would get to those labs and then they would discover the penicillin and then they would use it for their troops. So they tried to devise a way that they would be able to destroy all the penicillin except some of it that they would rub inside their coats. And then they would be captured or whatever, and nobody would understand what that was, but they were hoping someday they could take that and then regrow this strain of penicillin. But anyway, it's an extremely interesting book about scientists really sharing information and trying to greatly help the war effort. All right, so this whole idea of having antibiotics, they are really classified as antimicrobials because it's going to be anything with a small organism. And we have some things, some different terms. So we talked about antibiotic, and we'll talk about it down here again. But semi-synthesis is where you actually take a synthetic drug and then you synthesize it over and over again. So it is really not an antibiotic. It's not made by an organism, but it's something that's synthesized in the lab. And then the synthetic ones are just the antimicrobials that are completely started in the lab. So these up here, there's some chemical from maybe some antibiotic, and then they keep altering it, trying to make it better. So that would be a semi-synthetic, and this would be a totally synthetic. Just they dream up some molecule and then just keep testing it. And then we have the true antibiotics, and these are the ones made by an organism and to kill other organisms. And these drugs, they're probably the largest array of antimicrobials that we have, and these are like antibiotics. But we know now that there's fewer and fewer antibiotics that work, fewer and fewer that are being discovered, and so it's really a place kind of havoc now because most of the bacteria, or many of the bacteria, are becoming resistant to them. And then we have even fewer antiviral drugs, so drugs against viruses. Although since this slide was made, or since this PowerPoint was made, we've gone to the mRNA vaccines, and now they are really within 8 months or something, they had a vaccine made for COVID. So it's not really an issue. It's not that we have fewer now. It's just that we really have fewer ones that we need to make drugs for. So it's you almost feel safer getting a virus than you get a bacterial infection. Okay, so what do these microbes do with these antibiotics? Why are they even making them to start with? Microbes save their genetic material for production of their own genes that they need. But these are one of the things that in order to survive, a bacteria has to have some defenses. You know, they can't walk away, they can't swim away. Whatever area that they're in, they need to stay in there and they need to be able to survive with whatever organism is in there with them. So they use it, at least what science thinks is these antibiotics are created by bacteria as a defense mechanism. So you can see here would be some bacteria, and they can use them as maybe a deterrent for other bacteria, kind of scare the other bacteria away. They can use it as some kind of signal. So that signal could be a good signal, like let's band together, let's have quorum sensing or something like that. Or it could just be a signal that you need to get out of here, this is my territory. Or here we have some other kind of signaling molecule, and this could be something like maybe a quorum sensing or biological molecule that would tell the other bacteria nearby, let's do something all together to be more powerful. Or they could just use it purely to chase away other bacteria. This is a signal to them, this is my area. They don't really know what it is, but it triggers something. And the search for antibiotics, this is pretty old. You can see that we started early on in the 20s, and all of a sudden we started finding some antibiotics. You can see some antibiotics here listed, very small, none that you need to know. But the idea is that we started early, found a lot, thought this was a heyday. We thought it was the time that bacterial infections would ever kill anyone anymore. We'd always be able to find some kind of antibiotic. And then all of a sudden it started tapering off, and then all of a sudden resistance happened. And they started seeing that more and more drugs that we had were resistant, or had bacteria that were resistant to them. So now it seems like in the recent past we have not found many antibiotics at all. And the ones that we have found have been tied up in pharmaceutical companies. Not wanting to spend the money to make those antibiotics, they can make much more money someplace else. And it's not the greed necessarily of the pharmaceutical companies, it's the fact that they can get sued so easily. If they go through all the trials and then somebody has a problem with one of the drugs that they produce, especially an antibiotic, then all that effort that they put in to make that antibiotic could be destroyed. So they don't put a lot of money into it, they just don't feel they're going to get a lot of money out. So anyway, we need some new approaches. We have we have methicillin-resistant bacteria, we have multi-drug resistant bacteria, and then we have extensively drug-resistant bacteria. So it is that bacteria are starting to be resistant to every single drug that we have. So we need to find something else. And I'm just going to give you 2 ideas of new choices that people have looked for. And here's kind of our old approach. We would just go out and get some soil, kind of like we did at our very beginning for our environmental. You didn't go necessarily get soil, but you went and got some dirt and dust or something from someplace, and then you plated it on a plate, and then you looked to see how many colonies. And sometimes you didn't get any colonies, or you got very few colonies. So it's bacteria are very finicky. There's only about 1.5 or 2% of all the bacteria in the world that can be cultured. So we really, when we do these plates, we think that we have a lot of bacteria, but it's not as much bacteria there is out in the world. So anyway, this is usually what we get, and they check all this, and there's not new antibiotics here. So these guys down here, they made this thing called an iChip, and this was in 1985, and they just took this little device, and you can see how small the device is. What they felt is the reason that we can't culture bacteria, like we take a swab, we take some dirt, we spread it on a plate, that the reason the bacteria don't grow is because they're not in their home environment. There are not signals from whatever is in that same soil that they are surviving in. They're not receiving those signals, so they don't know that they should be able to grow. So they made this little thing called an iChip, And it's really hard to see how very small it is. This is a much enlarged portion. And these are little pieces of agar in here. So these are the plates that they would chip this out and then spread them. So this is then put into the ground, and once the bacteria is just about big enough for one bacteria to get each of those little holes, and then they could capture that bacteria, and that bacteria then would be able to get all the signals from all the organisms that are around it in that particular soil where it is. And they got about 50 to 100 antibiotics from doing this. So they had thousands and thousands of organisms, tested all of them, and then 50 to maybe 100 antibiotics. Right away, about 50 of them were toxic to humans, so they didn't pursue those anymore. Then they started pursuing the 50 that they had, and they found 3 that were really good. And now they have gone on with just 1. And this started in 1985, and they're still going through the processing part to get this antibiotic out there. And this is, it's called teixebockton, and it says a new antibiotic, but I think this was published in something like 1985. What this does, this is what the molecule looks like, so this is what the antibiotic looks like, and what it targets is, I'll show you on a slide when we talk about the gram-negative and gram-positive bacteria. On the gram-positive bacteria, there's this thing called teichoic acid. I don't know if you've seen it in any pictures of the cell walls, but they have this long red-looking thing, and it is it's kind of like an acid that sticks up, and it's a signal for other bacteria to recognize that this bacteria is whole. That is, there's nothing wrong with it. It doesn't need to be killed. So autolysis is something that bacteria do to itself when they think that there's something wrong with it and it will die. And teichoic acid is one of the signals to tell the cell you're okay. So they targeted, or this drug targets this teichoic acid on cells, and so the cell thinks there's something wrong with it and it will kill itself. So anyway, it's a good antibiotic, and it does gram-positive, Staphylococcus aureus, a lot of these things that we don't have antibiotics productively for. So it should be something that's produced in the marketplace, but it still is in trials. So it's really disappointing. Anyway, so this is one new avenue that they had. They just developed this new little chip. They still went after soil where we have found almost all the bacteria before. So they said, we still think that there might be bacteria there. And then another individual, Craig Venter, I don't know if anybody's ever heard of him. He was involved in the Human Genome Project, but not where people invited him to. He was a businessman, and he said, gee, we've taken the Human Genome Project, it's supposed to be 10 years. This is ridiculous. It should take maybe a year. And of course, scientists said, no, we have this way of doing things. He goes, well, I also have a way, and I will produce exactly the same stuff you do, but in about 1/3 of the time. So they wouldn't let him in. They wouldn't give him any human genome sequences to start genomes to sequence. And so Bill Clinton, when he was president, he came in and said, we have to play nice with each other. Here's a person who has all the technology to do it, and you have all the science to do it. You just need to share. So they started sharing with him, and within about a year and a half, the Human Genome Project was done. So it was purely because he had the technology in order to sequence tiny bit of pieces at a time and then group them all together. So anyway, after that, instead of being welcomed into the science community, he was still shunned because nobody likes to be outbid by a business person. They want a scientist to be able to have sequence this themselves. So anyway, what he did is he outfitted a boat, and then he said, there must be more antibiotics someplace. We're trying everything on land. Let's go try things in the sea. So he tried all kinds of putting materials into the sea. We got out the water, and then he tried to sequence everything that he had. And he didn't get any antibiotic discoveries at this point in time when I just checked this probably about maybe 5 or 6 months ago. So he hasn't given up. He's in San Diego now. He has a pharmaceutical kind of a company that does a lot of drug discovery. So anyway, that's the way that we are still trying to look for antibiotics, but you can see if we had a big breakthrough, it would be all over the news. But so far, nothing. All right, so here's another one, little clicker question we could do to kind of see if we understand this point. So who do we think? Without harming the patient. What's the name of that guy? Okay, so I see some A's. Everybody wants to give Pasteur the credit for everything, but it's really error. So I saw a lot of D's also. So yep, he is the one, or A and C, huh? But yes, so this would be the one. He's the one who selectively, first person to envision the use of chemicals. All right, so chemotherapy index. So when, it's still taking a long time to get this teixeboxam antibiotic through the system. And so really, what is the system? One of the things that they do is this thing called a therapeutic, chemotherapeutic index. And what they do is they weigh the toxicity to the human versus the efficiency of the drug to kill an organism. And they weigh those two numbers. And the idea, of course, they want to have the amount that's going to be toxic to a human to be very high. And they want the amount of the drug that's going to kill the bacteria to be very low. So that way, as a human, if you are taking antibiotics and you forget that you just took 3 pills, and you go and take 3 more, and then you take a nap and you think, oh, this is the next day, I need to take more pills, and you keep taking more and more and more, you can take an awful lot. If this number is very, very high and this number is very low, you can take an awful lot before it becomes toxic to yourself. So they want to make sure that that threshold between the two is a very big number, okay? Because they always take the therapeutic index is the toxic dose, what's toxic to the human, and the therapeutic dose. So if that number is a big gap, then they know somebody could not take too many and then harm themselves. Okay, so does that kind of make sense? So this is one of the ways they determine whether a drug is good to put out on the market. Okay, so here's another little eye clicker. So the therapeutic index, which one of these numbers would be the best? So this would be our therapeutic index. So we are saying, okay, most people saying, are you getting 2 answers? Or 1 answer, okay. So yes, A would be the answer. We want the biggest number possible. Remember, we want this number up here to be really high. So that the toxic dose to a person, you would have to have an awful lot of drug to kill yourself, and then only a small amount of drug to kill the bacteria. So you can take pill after pill after pill after pill, and it's still not going to be built up in the system and be harmful. So therapeutic index, you want a high number. Okay, so these are for antibiotics. There are going to be some antibiotics in here I'm going to ask you to remember. But these right now, this is just kind of an overall brush of what antibiotic spectrums are. So these drugs, they have a wide range of efficacy. The broad spectrum drugs seem to be the best, or logically would seem like they're the best to take. But we know that that is not better, because if you are taking a broad spectrum antibiotic, you're really killing your own microbiota besides maybe the target that you have and lots of other bacteria. You want to be as very specific as possible towards the bacteria that you are killing. So here are some that kind of have narrower range, like this is very, very narrow in here. Here's more of a broad spectrum. So this means you're killing a lot of other bacteria that you don't need to. And some of those bacteria then become resistant, it could be, to this particular drug that you have. So the idea that you really want to have a narrow spectrum, those would be the ones that would be targeted to as few organisms as possible. But generally what happens is you're prescribed a broad spectrum just so that you can get something started right away. And then maybe after different tests are done, then they can fine-tune whatever antibiotic you should take to more specific for the disease that you have. So that's all. Nothing that you have to really memorize on here. Just understand the difference between a broad spectrum and a narrow spectrum, and maybe why it's better to have one than the other. And one other thing I want to put on here is gram-negative bacteria have this lipopolysaccharide on their outer cover, and they have these pores in order to get in. If you're a gram-negative, remember you have the outer membrane. And that means that anything that has to get into the any drug that has to get in to harm that bacteria other than its cell wall on the outside or its cell membrane on the outside, it has to go through these little porins, or it has to diffuse through the membrane. And you saw textulobactin, it was a huge molecule that can't really diffuse through a membrane. So it has to be targeting something else. Okay, and then this is another little eye clicker really quick we can do. So this isn't something we talked about, it's more like a logic thing that you would have to think through. Okay, which of the following does not describe the ideal antimicrobial agent? Yeah, you don't want it in the body all the time. You want to prescribe it for a person for a limited amount of time. You want it to do its job, and then you don't want it to linger. So yes, A is a good answer. All right, these eukaryotic pathogens, what happens when there are those kind of organisms? What we've talked about just recently now is bacteria. So you can have something that targets the bacteria, but the bacteria don't have many of the things that we have. So you can target those things on the bacteria and not be targeting yourself. But when you're targeting organisms that are eukaryote, have the same kind of plasma membrane, they have the same kind of ribosomes, they have a lot of the things that are exactly the same as we do, then it gets much more difficult to find a drug that is going to target only that organism and not everything in our body. So that is much more difficult to find. But we're not really going to talk about that much. We're going to be just looking at the ones for bacterial. Okay, so, but not many things in the arsenal to be able to target eukaryotic organisms. So what we want to look at now is mechanisms of the antimicrobial action. How do they actually target a bacteria and what do they do? We know a bacteria is, I don't want to say it's a simple organism, but it has, you know, an outer membrane, it has an inner membrane, it does have a ribosome, it doesn't have a nucleus, it does have DNA, it has RNA, it does some of the processes that we do, it has some pumps on its membranes, but there's just not that many places that we could actually target. So we're going to look at the different ones. And this is, these are some, and this is actually from the textbook. I have a different figure I'm also going to show you. But this kind of shows you that here's a membrane. If you can look at this, here's an outer membrane, and there's a little tiny bit of peptidoglycan here, outer peptidoglycan, and then that little tiny membrane that's right here. So this must be a gram-positive, right? And then we have here where these are ribosomes, and they're making proteins. So this is something we can target. This is the disruption of the cytoplasmic membrane. Both gram-positive and gram-negative also have an inner membrane that's right next to the cytoplasm. So that is something that can be targeted. Just the metabolic pathways that the organism has. And this is when they inhibit like DNA and RNA synthesis. So this is the replication that we talked about the last time of the DNA. And then here is some attachments. Sometimes bacteria have to attach to the host in order to get into the host. So if you can figure out those attachment methods, we also can target those. But there's just not that many places that are able to target. And then, of course, we have to have the right antibiotic to target those regions. And if bacteria have not already made an antibiotic for us to mimic, to use against other bacteria, then we're kind of out of luck. So this is in the textbook, and I'm just going to be asking questions about these major areas. And this is a more simplified one. This gives you the actual antibiotics. The other one does too, but they're very small here. This gives you the actual antibiotics that are used for each of these places that are on the bacteria. So it's kind of the same thing. Here's the cell wall they're pointing to. Here's the nucleic acid. Here's protein synthesis. Here's a ribosome. Here's a membrane. And then here is folic acid. This is a process that the organism does. So this isn't all-encompassing. This isn't everything. But some of the checked ones here we will look at individually to see the antibiotics. And I believe penicillin, cephalosporin, maybe chloranthinicol, tetracycline. These are the antibiotics that we're going to use in lab tomorrow. So we'll actually see these and we'll see them in action on different bacteria. Yes?
So you want us to memorize the ones that are checked?
Yes. So there'll be things that we talk about later on that means that's the antibiotic that you'll want to focus on when we talk about that. Okay, so I'll have something on each of these that will tie that into the lecture. Okay. So first, inhibiting DNA synthesis. So we're looking at this point down here. This is something that is involved with RNA and DNA synthesis. So we're going to be looking at this folic acid. So this is the sulfamides that we're going to talk about. Okay, so for this one, it kind of goes back to some of the other things that we've talked about. Here's broad spectrum, so that's what kind of antibiotic this would be. Because it targets making RNA and DNA, and all organisms, no matter whether they're gram-positive or gram-negative, will make RNA and DNA. This means that when you take this antibiotic, you're doing both gram-positive and gram-negative bacteria. There's no differentiation between them. And folic acid is a kind of like a vitamin or a cofactor that's used by bacteria. They have to synthesize their folic acid that they use in making a DNA and RNA. We get it from our diet, so this isn't a harmful drug for us because we don't really care about it. We already get it from our diet. But bacteria need this folic acid. So this was a target that sulfur drugs would actually interrupt the ability for this molecule to be able to be this folic acid to be made. And so they use competitive inhibition by outcompeting. This is the molecule that they made, and they outcompeted this molecule. And you can see how similar they look, just not exactly the same. But this molecule will fit in the space that this one should feed in. Just by taking enough of it, it outcompetes. And if this one fits in the space, as I'll show you in the next slide, it means that the folic acid can't be made. So here's kind of a quick little mechanism of it. Here are the two look-alike molecules. This is what's needed, this PABA, and this is the one that's substituted as a drug. So here's what normally happens. This is the enzyme. You don't have to know the enzyme, and you don't really have to know the whole mechanism, but you can just know that this is just a competitive inhibitor, and it won't make folic acid. So here are just some molecules come into the enzyme. Here's the PABA that comes into the enzyme. When all three of the reagents are in, it synthesizes folic acid. So that's exactly what the bacteria need. So now a new drug was made, a sulfur drug that actually outcompetes the PABA from getting into the spot. So now this is in there. It doesn't quite fit. There's got a little extra thing here. It just won't fit the other ones in the active site. So it inhibits the enzyme from doing its job. It can't make that folic acid. Yes?
How does it outcompete with it? Like, is there more produced than the PABA, or?
Yes, so the organism has to produce the PABA, and we synthesize it, and we give a patient a whole bunch of it. Mm-hmm. So it's just, there's not enough of this to outcompete. Oh. Yep, so it's really a competitive inhibitor. Okay, so that's one way that we can just prevent this. Except, I think we've talked before that bacteria are very, very clever. You can't really outthink a bacteria. You can't say, okay, we blocked this path. You might have blocked it just for a very short period of time. Because bacteria have some probably mechanism within their arsenal that they can call out and then start using when something is blocked. And that's exactly what happened. So these sulfamide drugs don't work anymore, and the reason they don't is because there's a substitute pathway now. So the organism no longer makes folic acid. So what it does is it says, okay, humans get it from the outside, I'll just get it from the same way. So they use a whole other pathway, and now they're able to absorb the folic acid right from its environment. So very, very clever, and it happened in a very short period of time, like a year or two. Anyway, so now what they do is they try to make synergism. That means they use a couple of different drugs to hit the bacteria at the same time, and that way the bacteria cannot become immune to it. All right, so drug combinations, that's the biggest thing that they can do. Try to use several different drugs at a time. It's just like we do with viruses. We try to get several different things that hit that virus at the same time. so that the vira cannot continue to operate. All right, so here's another quick little eye clicker, and these are all ones that you could definitely see on a quiz or an exam again. So kind of quick things, easy to remember now because you just saw the information, but maybe harder when you see it much later on. Okay, so we're saying this is going to be okay, all right, yeah, so people change their mind. Perfect. D is the one. So it is competitive like this, but it doesn't synthesize PABA. PABA is one made by the organism. It is outcompetes so that folic acid can't be made. Okay, good. So I think a couple of you had B, and then you change it to D, so that's perfect. All right, so now that's that. So there's one drug for that. That's one of the things that bacteria do. So cell wall synthesis, this is pretty important. So we'll also talk a little bit about membrane. So this is the exact same slide again. So I want to talk about these two antibiotics and then a little tiny bit about this one. Penicillin, it's going to target the peptidoglycan. You probably recognize this as the gram-positive cell wall. Here's all the peptidoglycan up here. Here's the teichoic acid that I've mentioned before. That usually sticks up a little bit above the cell, so that's when autolysins can come by and look at the cell and say, oh, you're doing okay, you still have your teichoic acid. And that's what that previous antibiotic did. So anyway, this is the way these cell walls are made, the peptidoglycan. So these are really made of sugars. That's not a shape of a sugar, but each of these are sugars. And the two sugars that we use are N-acetylglucosamine and N-acetylmuric acid. You don't need to know the full name, but you should know, or you should be able to recognize the enzymes or the abbreviations. So you can know what those mean, that they are the sugars that are involved in the peptidoglycan. So we have something called peptidoglycan. So this is the glycan part right here, N-acetylglucosamine and N-acetylmuric acid. And these are the peptide part. So these are small little amino acids, and they are like chains that lock this together. Okay, so like you think of a log cabin, this keeps the cell wall outside the bacteria and stable. All right, so how do drugs affect these? So here's one way. Again, you have to recognize this. This is a little bit more definitive. So this is a gram-positive, and this would be a gram-negative. So you can see here, if we are going to inhibit the production of the peptidoglycan, this peptidoglycan is very, very exposed to any antibiotic that comes in here. This one, the antibiotic has to go through the membrane, and then we have just a little bit of peptidoglycan that it could destroy, and then if they had to get into the cell, it'd have to go to another membrane. So gram-negative are really not affected by these antibiotics that I'm going to talk about. There is some effect, but not a lot, and we'll see that in when we do our analysis in the lab. So the mechanism of action of penicillin, this is a molecule from the bacteria. It's called transpeptidase. And what transpeptidase does is you can see it right here. It kind of comes in, and you see these little arms that are sticking out. If you think of a log cabin and you're building the log cabin like this, you're putting the logs on top, you have to have some cement or something between the logs or they'll just fall apart. So these are the cement between the two logs. And they're not cemented together until these two yellow arms are locked together. So you can see here, like this one is locked right here, but these are not yet locked. So transpeptidase comes and binds them, and then it switches one over and locks it together, and that makes sure the logs can't fall apart. So transpeptidase is a target. So we have penicillin, and penicillin looks like this in a very stylized form. Penicillin fits right into that active site of the transpeptidase. So transpeptidase now has a penicillin molecule lodged inside of the active site, and now it cannot go and cross-link the peptidoglycan on bacteria. Okay, does that kind of make sense? So transpeptidase is an enzyme from the bacteria that makes its cell wall, or at least cross-links its cell wall. penicillin binds in the active site of the transpeptidase, and it inhibits the ability for transpeptidase to operate. And therefore, the cell wall won't be made. So taking antibiotic, this particular antibiotic, penicillin, it's very important to take it right when the cell is going through that exponential growth, because that's when it's making their membrane. If your membrane and your cell wall is already made, the peptidoglycan, you can take this all day long and transpeptidase isn't active. So it really doesn't help. So it only helps when the bacteria are actually making their cell wall, cross-linking it. And so we want to take the antibiotic when cells are actively dividing. All right, so here's one mechanism. And then this penicillin, there's various versions of the penicillin, so I'll show you on another slide. It has a very high therapeutic index, so that is good. You can take a lot of it and it's not going to hurt the human. It's active against gram-positive, and we can see why. There's all the peptidoglycan on the outside. The molecule does not have to get into the cell, it just has to get to the outside of the cell, and that peptidoglycan is exposed. It will do some gram-negative, but not very many. And that's obvious to see. Would it have to go through a membrane on the outside to get to the tiny bit of peptidoglycan that the cell has? And since it interferes with the cell wall synthesis, osmotic pressure will cause the cell to start to burst. Either water will come in, or water and then cause it to burst, or it will then shrink down. Either thing that happens with osmotic pressure. And it's most effective against rapidly growing bacteria. So at that time, it's bactericidal. So this is a term that you should know. So cidal means it kills the bacteria, and that as they're rapidly dividing, the cell wall is not made, or the peptidoglycan, then obviously it's going to kill the bacteria. Down here, if they're multiplying very slowly or they're pretty dormant, then it's bacteriostatic. It just won't allow the bacteria to grow anymore, but it won't kill the existing bacteria that are there. So in this case, if the bacteria are growing very slowly or they're dormant, then this becomes static. There's just no effect on those bacteria. So it just keeps everything at bay. So two terms that you should know. Okay. And then this is penicillin. You notice this ring right here in penicillin. This is a beta-lactam ring. And this is where the penicillin A's, that's made by the bacteria, it will come in and cleave this. And when it cleaves it, it destroys the antibiotic. So this pink form right here is the same as the same little pink forms here. And you can see that we've had natural penicillin for years and years. And when it was no longer effective, science came in, we tried to mask it. We realized that this was the part, this part right here was the beta-lactam ring that was being cleaved by bacteria. So they tried to make extensions that came out and then would fold down underneath and then protect this site so that penicillin A's or other beta-lactams could not go in and cleave it. And so you can see all kinds of different molecules have been made with overhangs and things, trying to get an effective antibiotic. But right now, it's not as effective as it was. It's still a good drug, but there are still lots and lots of bacteria that are resistant to it. And then what they now have done is they've taken, especially amoxicillin and amphotericillin, they've taken it and they give it two drugs at the same time. So you take your antibiotic and you take another drug. And you might have noticed after I learned this, I started looking at the drugs. I have a lot of pets, and those pets take different antibiotics. And I noticed now every time I get ampicillin from them, they always have this clavulanic acid with it. So what that is, let me go to another slide. This is what the okay, I didn't put it here. Oh, it's just saying that penicillin A's will cleave right here that I was just showing on this slide. And you can see, I can ask you what penicillin A's uses to cleave this, this beta-lactam ring. What does it use to help in the cleavage? Water, right? So you can see what happens when this is cleaved. You get an OH on this part, OH on this part and an H on this part, and this is how it cleaves it. So you know it's using a water molecule as far as part of its mechanism of action. So, okay, and so each antibiotic will have a specific beta-lactamase. This is just a general term of all the beta-lactamases that are out there that cleave beta-lactam rings. So this is not the only one. There's other antibiotics that have it also. So to protect this, this is where they've actually taken amoxicillin. This is what amoxicillin looks like. You can see it's a beta-lactam ring right here. So you should be able to recognize a beta-lactam ring. So if you have a drawing, you should be able to see it. And I wouldn't mask it in any way. It would be as plain as this. So what they do is they take amoxicillin, and this is the same kind of thing, and this tazobactam. So you look at these two together, and this is what they feed you together. So this one is piperacillin. I got them mixed up on that slide. So this is the one that is clavulanic acid, and that's with amoxicillin. So these should be reversed, and these would be the little vials that you get. And I think that's what we get. We get, I think we get amoxicillin, and I think it also says it has clavulanic acid with it. So what happens is you can see this is the clavulanic acid, and you see it also has a beta-lactam ring. So if the organism is producing a beta-lactamase, whether it's a penicillinase or whatever it is, and you give them two different things to cleave at a time, it's going to be these two. When you give it two different things to cleave at a time, it's going to absorb a lot of that beta-lactamase that is just going to hit a target that's not the antibiotic. So the antibiotic can still be effective. So and this is a harmless drug. It doesn't hurt at all. So you can give it in 10 times or 100 times, well, that's exaggerated maybe, at least 10 times the dose that you have for your antibiotic. And so you have a whole bunch of molecules for the beta-lactamase to start cleaving the ring, and it will leave the amoxicillin alone. Yes?
Do all bacteria have beta-lactam rings?
So this would be antibiotics.
Do all antibiotics, I meant, I'm sorry.
Oh, no. There's about 30 that I know of from the antibiotics that we have, and that's even with derivatives of the same thing. Because you saw all those ones for penicillin, a whole bunch of derivatives. They all have beta-lactam rings. And there's going to be 2 more that I show you do. So you'll be able to recognize that beta-lactam ring here.
So does that mean all of those antibiotics with beta-lactam rings have to have something like clavulanic acid to?
Some of them are not, a molecule is a certain shape, and let's say the beta-lactam ring is right here, but if they have something like this, and the enzyme that cleaves beta-lactam ring can't get through that little opening to that part of the antibiotic, then it's safe. Yeah, so there's lots of things. That's what they tried to do with penicillin. They tried to design more wings on it. The penicillin part was the same, but the things that they put on the outside was to keep the beta-lactamase, because it's a big enzyme, to get in there and cleave that little beta-lactam ring. But it doesn't work. And months, even in fact, 4 or 5 months after they made a whole nother synthesis, it didn't work anymore. But anyway, so they tried this. And this is just a little bit easier, but you don't see this all the time. Every time you get a penicillin or those type of drugs, you don't see an extra molecule with it. So this is not prevalent. But these are just 2 that I'm aware of because I've actually seen them for my pets. Okay, so this is called synergism, then they're just putting 2 together. So here's another cephalosporin. So here we have a beta-lactam ring. Okay, and so here's another one. We have a beta-lactam ring. So there are a lot of beta-lactam rings out there, and any beta-lactamase is going to be able to get in there and destroy it. Okay, so this is the only thing I wanted to show you. This is just another beta-lactam antibiotic, and just so you can see this on here. So we're not going to talk about it, but it has cephalosporinase, just like penicillin has penicillinase, and that just means those are the ones that cleave the beta-lactam ring for that particular antibiotic. Okay. So, this antibiotic, the only thing I would do is show you a series of these things, and you would point out that they have a beta-lactam ring. Okay, so this is another one I would like you to know. This is vancomycin. This used to be the drug of choice because they were holding it off the market. They only gave it a few times, but it took only like 5 months after it was used a little bit more regularly that we had resistant organisms. So it was kind of our one of our antibiotic that would be our last resort, and it totally failed with that. You can see how very large it is. And I use this molecule a lot to have students count the number of sugars. So if you just looked at this, you could obviously, these are sugars because it has sugar in the ring. So you look everywhere else, and always if you're trying to look for a sugar, you're looking for a sugar, an oxygen in the ring. So these are not, these are like peptides, but this is a sugar. Like towards the final exam and stuff, I would show you a molecule, and you would have to pick out the sugars on it, different functional groups that we had. And then here's another semi-synthetic vancomycin, because this was becoming resistant, or was developing, bacteria were developing resistance to it. So they synthesize another one like this, and it's also, it's not really prevalent anymore. I don't even see it on the market. It either didn't work or it did become resistant quicker. Okay, so this is a glycopeptide. So this is a peptide here, and then this is just a sugar up here. So vancomycin targets the cell wall, and so I'm going to show you that. So here is the mechanism action of vancomycin, a little bit different, same cell wall, so it has a different mechanism. So here is transpeptidase, like we have seen before, and we see what transpeptidase is going to do. Well, vancomycin doesn't really harm transpeptidase. You can see it's not interacting with it at all. What it does is it comes and clamps around these parts right here. So when transpeptidase comes to actually cross-link them, it can't, because it has vancomycin already clamped around the peptides. So this one has a totally different mechanism of action, but it's the same place on the peptidoglycan as a cell wall. Okay, so it just inhibits the transpeptidase from cross-linking. It's not a competitor. It's not. All it does is it blocks it because it's already got to the site before. And so, of course, I said that vancomycin wasn't so great anymore, and so this is within, I don't know, it was like 5 months. The organism, you can see here there's a certain series of amino acids. Up here, you can see there's a certain series of amino acids, and down here you can see there's something different. We have a different amino acid at the end here, kind of a square amino acid. It's not square, but it has different R groups. And so when the vancomycin went to grab onto it, like it normally does, it doesn't fit anymore. It doesn't fit in the spot that vancomycin has. So this means that just having the organism change one of its amino acids to be something different now allows it to avoid being attacked by vancomycin. So pretty amazing that the organism can just do that. And this is just DNA synthesis. So DNA changed the nucleotides, so now just put a new molecule there. Okay. So anyway, this is vancomycin, so I do want you to know this kind of a mechanism, not to draw, but understand there's a difference between this mechanism and the mechanism back, this isn't go back very well, the mechanism right here. So there's a difference between what's happening to transpeptidase in those two mechanisms. Yes?
In the mechanism of action of vancomycin image,
Okay.
is the thing that's changed, did they add or added the lactate?
So I'm sorry, say that again?
For it to be resistant, it added the lactate, that purple stuff.
Yes, yes.
But it could add any one of those in the key.
Well, these here are what were already there, and it just changed it to something different. And so this is just probably a much bigger molecule, so that when the vancomycin tried to clamp around it, it just doesn't fit anymore.
Is it always gonna be D-lactate, or is it?
Well, the organism has to have already designed that, and so now that's in its way to build its cell walls.
Okay.
So one of the bacteria must have had a mutation, had been using this all its little life, but it was never a problem. And now it's a problem because this vancomycin came along, but it wasn't a problem for that particular bacteria. All the other bacteria were being killed, but this one was not. So now this one survived because it has a mutation. And that's really what that is, one little change like it. It's not like it tried to figure out how to get around vancomycin. It's just that it already had a mutation there, and now that bacteria now has outcompeted. All right, so we'll continue on until it's maybe a little too tiring. And then the only thing I can do, because we have to go on from this particular lecture to another one when we meet next week. We can't dally on this. So these are posted, so then you will just have to go through the rest of it on your own. So I'll try to go through as much of it as I can, as quickly as I can. But this is a chapter that matches our lab now, and then when we go to the next lab, we have a whole other lecture we have to do for that. Okay, so this one is on polymyxin, and this is really for topical. So polymyxin is for membranes, and every membrane, whether you're a bacteria or whether you are a human, it all has that same membrane where you have a lipid and you have a phosphate head group, etc. So that making of a plasma membrane is very, very similar between bacteria and humans. So we can't use this. We use polymyxin as a topical. So if you have something on your skin or something and you're trying to kill the bacteria, they're fine, because it also kills your own skin, but most of the skin, about 6 or so layers deep, are dead anyway. So this doesn't really cause many problems. But this is one of the ones that we can use, and it's purely topical. Okay, so this is one that we can use for membranes. So we've covered pretty much the peptide glycan, and now we've covered the membrane. And you can also see how very big polymyxin is. It's a huge, huge, huge molecule. So polymyxin is one of the ones I want you to know that would be for membranes. And then vancomycin and penicillin would be the ones for the peptide glycan, the cell wall. Yes?
How does it insert itself into the membrane if it's so big? The polymyxin?
Oh, you see this little part right here?
Oh, that's what it is?
Yeah.
Oh, I thought it was the whole.
Oh, well, no, this is the whole molecule.
Oh.
This is the business part of it, but this is the way it gets into the membrane.
Oh.
Yeah, it hooks itself into the membrane. Yeah, and then it just destroys the membrane. And I don't know how long this is. That R indicates that it's much, much longer. Okay, and then not this one. We're not really going to talk about that. Okay, and so for this mechanism is what? To inhibit the formation of the peptide glycan layer in the bacterial cell wall. So one we know for sure, right? So what does cephalosporin do? So we can go back and look at that. That's part of, it doesn't actually, well, it's involved in the cell wall. Penicillin's involved in the cell wall, and these beta-lactam. These are like the rings that are involved. So the mechanism of what is to inhibit the formation of the peptide glycan. Yep, so I think definitely penicillin does. When you think of the mechanism of penicillin does, cephalosporin would also do the same thing, and beta-lactams also do the same thing, the ring. So D would be the best. It's all of those. Okay, make sense? All the ones we talked about. Okay, so now let's just do ribosomes and protein synthesis. So ribosomes, these are these little things right here. And aminoglycosides, chloramphenicol and tetracycline. I believe these two that we use in the lab on Thursday. So anyway, this is what we're targeting. This means these antibiotics have to get inside the cell. So they have to go through the outer membrane and the inner membrane if it's a gram-negative, and they have to go through the peptidoglycan and the inner membrane if it is a gram-positive. So that means these antibiotics, and they are very, very large, they still have to have a way to get in. And that's usually pores that are in the membrane. All right, so protein synthesis, we've seen this before. This is a ribosome, and we've seen it before. We know bacteria are 50S, or ADS, and or 70S, sorry, and then we are ADS. Again, they don't add up to what we would think. So here's a 70S ribosome, which is bacterial. The aminoglycosides, and amino is just amino acids and glycoside sugars. So these are the ones, and they're both going to target the 30S, and tetracycline also targets the 30S, and then chloramphenicol actually targets the 50S. So we have a couple of antibiotics that are used to target the ability for a bacterial organism to synthesize its own genetic material. And unfortunately, I think I mentioned that some of the ribosomes that are in our mitochondria are 70S because they mimic what bacterial are. So the rest of our body has 80S, so the 70S are in the mitochondria. And the mitochondria genes are not totally making only things for the mitochondria. They make a portion of other things, things to do with auditory, vision, a couple of other things like that. So they want to make sure that young people, babies to 5 years old, developmental stage, do not take tetracycline or aminoglycosides. Because what will happen is it will actually damage the DNA and the RNA from the ribosome, from the mitochondria that come from the ribosome. So usually there's warnings that you don't take it if you're a certain age, or there's warnings if you are pregnant and you don't want to destroy any mechanisms that are going on in that fetus. Yes?
Would you say that certain classes of antibiotics have like more allergic reactions than others, and if so, why?
Ooh, there are some that have allergic reactions. Penicillin is obviously the very famous one. And it's usually not the penicillin molecule itself. Penicillin will bind to other molecules in the body as it's being processed, and that's what causes the allergic reaction. Because most people that are allergic to penicillin, they weren't allergic the first time. You know, they go to the dentist, they get penicillin or something, and then the next time they have an allergic reaction, they go, I've done this before, I don't know what happened now. So there's usually something else that triggers. It has to happen once in the body, and then you become allergic to it. And other antibiotics, I don't know. I've only been in the hospital once or twice in my life, and I had vancomycin. So a doctor that was going to treat me, I was in the emergency room, and the doctor who was going to treat me was late coming in. They had called him, and I heard him come in the door and stuff, and they said, okay, start the antibiotic. And so they gave me this antibiotic, and all of a sudden I could feel this hot, it was like a rash, and they were stop the antibiotic. They said, you must be allergic to vancomycin. I said, I've never even heard of vancomycin. I've never had anything. And so they said, well, this is very unusual. So I guess people do have allergic reactions to it. Mine was just a horrible rash. But yeah, so antibiotics, they're usually generally very, very safe. I know very few people that have ever had, only penicillin. Have you known other people, or?
I see a lot of, like, I work in a clinic, and I see a lot of people are allergic to sulfur drugs.
Ah, okay.
So I'm thinking, like, it's synthetic.
Yeah, I don't know, could be.
So I don't know.
Yeah, I don't know. Okay, So here's another thing I would want you to know that what's happening to the 30S and why the 70S in total could be affected by 2 young children and maybe pregnant women. Okay. So these are what amino glycosides look like. So amino, you're going to think of the amine group, and then you think the sugars. So this is what they look like. Streptomycin is one of them. You don't need to know this part. All you need to know is aminoglycosides bind to the 30S. So not this antibiotic nor this antibiotic. But you can just see how very big they are. They have to get inside the cell. This is not so big, but this is pretty big. And it's really hydrophilic also. So I'm really surprised it can get across the membrane. Okay, here's tetracycline. Again, this is another one that usually they don't have young children taking. You can't see it on this one much, but there's a big ridge around the top. I've seen a lot of people that take tetracycline and get that ridge on their teeth. So the only thing on this one is I just want you to know also that this is involved with the 30S. Okay, you don't have to know all of this stuff. You don't need to know what it looks like. Okay. And chloramphenicol, that's another one. This is a 50S. So we've got a couple for the 30S and we have one for the 50S. And these are all obviously going to hurt the ability for proteins to be made because it's the ribosome. Okay. I believe that chloramphenicol you might see tomorrow also. Okay, so let's look at this. Which of the following best describes the mechanism of action of aminoglycosides? This should be pretty easy. Just talked about it. But again, think of a question like this that you might see in 3 weeks or 4 weeks. And it would be harder to answer then. So easy to answer now, but harder then. So D is exactly the correct answer. It's ones like this that you will see. Well, guess what? I don't know what I pushed, but let me, we're really getting close to the time I think we should stop anyway. But let me just go through them, not that rapidly, but let's see what you need to be kind of looking at on your own. Let's see, reset it over here. I don't know what went crazy. Maybe that was okay, so this is, let's see. Okay, so there's not a lot more after this that we talked about, so I'm just going to go to each slide and kind of just mention it. Obviously nothing on here that you need to know, nothing on there that you need to know. So this is just going to tell you there's some tests that we're going to talk about. The one that we do in lab is a Kirby-Bauer, and we do a diffusion one. So this I wanted to say a little bit about is we are using antibiotic discs, and those antibiotic discs will have diffusion away from the center. I think I have a slide on this on a quicker question. So if you look at the disc, here's the agar down here, and you're putting the disc right here. And of course that disc has antibiotic impregnated in it. And so what it's going to do, in fact, I'll get 2 of them. Here's another plate right here. So here's the agar, here's the agar. This is going to go down like this, and it's going to go out sideways like this. So it's going to keep going like this, and this is how it diffuses out. Okay. This plate, you can see it's much thinner. So here is the antibiotic. It's going to go much further because it hits the bottom and goes out. So one of the things, and there's other things that we'll talk about, this may have a ring this big around it. This one may have a ring this big. So you can't look at the distance. You can't say that this is a better antibiotic than this particular one, because every antibiotic has a different size to it, and so the size going through agar is going to either go faster or slower. The depth of the agar also is going to make a difference, because if it hits the bottom and it starts going out, that would make a difference. So Kirby-Bauer is a uniform methodology that we will have tables that tell us exactly how big a ring is for a specific antibiotic and what that means. So we're going to say it'll tell us whether it's susceptible, whether it's resistant or not. So we'll measure, and each one will be a different antibiotic, and we'll measure it, and then it'll tell you what you need to know about it. So this Kirby-Bauer, things have to be uniform. These have to be a certain antibiotic. You buy the discs, and the discs are only certain concentrations because you can't compare one concentration to another. It has to have the exact same depth of every single plate to meet the standard for Kirby-Bauer. Otherwise, you can't use their list. It wouldn't make any sense to compare this, because you have big, thick plates, with this where you have a thin plate. So you have plates that are exactly match the standard that Kirby-Bauer has. You incubate it at the proper temperature, they tell you. The depth of the bacteria, you can see this bacteria is spread on here. We won't be spreading ours on. That has to be spread to a certain standard. There's usually a tube. We don't use this, but there's a tube of density, and you hold up the tube of density, and you make sure your bacteria that you're going to start to spread has exactly that same density. You don't use a spec for it, you just really hold it up. I suppose you could use a spec, but that's not usually what's done. So there's so many rules and regulations with Kirby-Bauer to make sure that you can use these forms to be able to determine how, whether the antibiotic that you have spread on the bacteria you're interested in is receptive to that antibiotic. Okay, so you get more on Kirby-Bauer in the lab. And this is called a disc diffusion method. So I'm going to skip this one, but this is, I'll leave it up just enough time for you to quickly read it, but you don't have to answer it. Okay. So again, good question that can be asked later on. This is a different kind of a test where you can tell exactly how much antibiotic is, the concentration of the antibiotic can actually stop the spread of the bacteria. The Kirby-Bauer method, that you can't do that. This has a whole bunch of gradations on it. The Kirby-Bauer does not. Okay, so a little different. We don't use that in lab. There could be a lot that we could talk about, but I really won't. This is just a tube dilution, and I think you can just read this on your own and kind of look at the graphs. It would take a little bit too long. This test I do want to say something about. This is for beta-lactam rings. They have to make sure that the organism that you have does not harbor a beta-lactam ring, beta-lactamase. So they take this molecule, and you can see that this molecule is all connected here. The circles just show the two ends of it. There's a beta-lactam ring right here in the middle. And that beta-lactam ring, if that beta-lactam ring is cleaved, what will happen is these two ends will fall apart, something like this on the bottom. And when they're held together close like this, with all these rings in them, they quench each other. They have these rings, and these rings will have electrons that delocalize around the rings, and then they will cause fluorescence. So if you keep them close together, that's what will happen. So if you cleave this, what will happen is as they move further and further apart, you'll quench the fluorescence. And then when it's, you'll quench the fluorescence over here because it's dark, and over here it's unquenched. So the idea is you want it, when you cleave this, you will see this, fluorescence that was previously quenched when they were closer together start to show. So this is a pretty common test that they do to see if the patient has a beta-lactamase. If it does, then you cannot give them any of those antibiotics that have beta-lactam rings. Okay, so it kind of heads off just giving antibiotics that aren't going to be effective. Okay, and then I think antibiotic resistance, we've talked about this before. You can read this on your own. I think this is pretty self-explanatory. Again, this just shows how antibiotic resistance happens. So again, I think pretty self-explanatory. This just shows and talks in detail how can a bacteria prevent the drugs that we give them. Sometimes they could just take it out, sometimes they could cut the beta-lactam ring. So these are the mechanisms that the bacteria uses to resist any antibiotic that we give them. And this is just a little summary of that. It talks about some of the antibiotics that are most resistant. And all of this is just statistics on saying that we need to find more antibiotics. And this is saying why some of them are, they're really inexpensive in some other countries, and they're also just going over to Mexico. You can get antibiotics for anything. You say, oh, I'm out of my antibiotics, I'll just go to Mexico and buy them. So of course that's not always the best way. And I think this is probably it. Okay, so this is just a little thing as what can we do, and things like this. Take a class like this, understand. And as we were kind of mentioning before, a lot of people will take their antibiotics only partially and not complete them. And what happens is then that causes some bacteria to become resistant to those antibiotics. So you have to make sure you take all the antibiotics. So that's really all this is kind of a wrap-up slide. Okay, and so that's it. So really, it's only the very last part there I kind of left you to look at it, and those are already posted, so you can go on to the website and look at those. But we will have to move on to another chapter next Thursday, and I don't really know what that is. Okay, so any questions or, no, set? All right, and everybody got 17? Or 17? Yes, perfect, thank you. Yes, you too. We're almost done. You too. Almost over.